Hello everyone. This is Dr. Atul Pradhan, Assistant Professor, School of Agriculture, Sage University, Bhopal. So after the concept or video lecture of the pre-Mendelian concepts of heredity, today we will discuss about the post-Mendelian concepts of heredity. So what are the post-Mendelian concepts of heredity, or what does it mean? So we all know that the basic principles of heredity were initially discovered by Mendel, and definitely they were rediscovered by these three scientists, Rice, Lorenz, and Sharma in 1900. After this, these three principles of Mendel, they were clarified and confirmed by several researchers, and on that basis, some new concepts were originated or investigated, and these concepts were also known as Mendelian deviations or Mendelian exceptions or Mendelian anomalies. As these concepts or Mendel has no idea about these concepts, that's why it is called as the exceptions or anomalies, and they are discovered or investigated after the Mendel era. So these are the list of various post-Mendelian concepts, just like incomplete or partial dominance, codominance, linkage, lethal genes, gene interactions, pleiotropic gene effects, polygenes, maternal effects, etc. So we will discuss one by one in brief about these concepts. What does it mean? What are the examples? How they are working? So let's start by this incomplete or partial dominance. So it defines that it is a form of gene interaction in which both the alleles of a gene at a locus are partially expressed. Means, if we observe in the given picture, that the parent has two colors, that is the white and red, and if we observe the F1 or the offspring or the uh, result from this process, we observe that the color changes to pink. That's why it is one type of interaction where the genes at a locus are partially expressed. Means in this F1, it some of the color from the male and some of the color from the female is expressing and they are mixed. That's why they are also called as the blended dominance. Blended means mixture of both the colors, and the phenotypic and the genotypic ratio are identical in the case of incomplete dominant, that is 1 is to 2 is to 1. And in the picture, we are observing that flower, the example that is the poroclock plant, that is the Mirabilis gelata. So this is about the incomplete or partial dominance. Means both are showing their expressions but partially or incompletely in the F. So this is for the first concept. In case of codominance, codominance means co means both and dominance means both are expressing their expressions. So when both the alleles express their phenotypes in heterozygotes, they are called as the codominance. The best example, that is the AB blood group in man. In case of AB blood group, the man having the AB blood group, they are having the both the alleles, both the genes of A blood group as well as B blood group. That's why codominance. Both are expressing themselves, A blood group as well as B blood. So in other words, heterozygotes for codominant alleles are phenotypically similar to both the parental phenotypes. So that is the codominance. If we want to observe the difference between the codominance and incomplete dominance, so in the given picture, in the type of fish, if we observe that both the parents having the blue color and red color, but in case of F1, both the color are expressing themselves, means both blue and red color, that is the codominance. And in case of incomplete dominance, if we observe the roses, so one of the rose is red, one of the rose is white, but in case of F1, we are observing the pink color, means both the colors are expressing, but in incomplete way or partial way. So this is about the codominance. Third one, that is the multiple alleles. So multiple alleles means number of alleles. So existence of more than two alleles for a gene is called as the multiple alleles or multiple allelism. The best example that is the ABO blood group alleles in human or the coat color in rabbit. So in this case, the gene has more than two allelic forms, although only two can exist in diploid cell at a time. In the given picture, if we observe the genotype A, blood group genotype A having the antigen A, or the blood group genotype A having the genotypes IA and IO. In case of B blood group, it is having the multiple alleles IB and IO. In case of B blood group, the same is having the IB IO as well as IB IB. In case of AB blood group, both are having the antigen IA and IB. And in case of O blood group, there is no antigen. So means here multiple alleles are working on the same platform. That is called as the multiple alleles. The fourth one, that is the linkage. So linkage means to link. That is the tendency of two or more genes to remain together in the same chromosome during inheritance is called as the linkage. So in the picture, it was clear observed that if the two genes are present closely to each other on a chromosome in the first picture, so it will be linked. That is called as a linkage. Similarly, if genes are situated separately on the two other chromosomes, so definitely just so in this second picture, if we observe that these two genes are situated separately on two other different chromosomes. So definitely there is no linkage. Also, similarly, in case of this single chromosome, if genes are far away situated from each other, so in that condition also linkage will not be there. So that's why this tendency of two or more genes remains together. So in this condition, they are remaining together in the same chromosome during inheritance. So that is the part of the linkage. So this linkage observed by Batson and Punnett in the P, Hutchinson in Mage, Morgan in Drosophila. So this is about the linkage. Next, that is concept that is about the lethal genes. Lethal means death. Means if the organism or any individual carrying that gene, it may leads to death. So gene which cause death of its carrier in homozygous condition. Definitely, the genes which are observing in the homozygous condition, it will leads to death. In presence of lethal genes, would be also called the Q0 genes. Okay, the normal segregation of 3:1 is modified to 2:1. Why? Because one of the genotype, one of the individual has to die. In the picture given here, so in the mice, allele of yellow coat color is dominant over agouti. So in the black, that is the agouti color, but yellow color, coat color, that is dominant. So in case of lethal genes, this fourth mice has the dominance yellow coat color. And definitely, the definition says that gene which cause death of its carrier when in homozygous condition. The fourth one, we are observing that the yellow coat color is in the homozygous condition. So definitely this has the presence of lethal gene. So it will die. So this is about the concept of lethal gene. Next, that is the gene interaction. We all know that genes are interacting with each other. So according to Mendel, they observed the ratio of 9 is to 3 is to 3 is to 1 in case of dihybrid cross. But maybe there may be chances of deviation in that dihybrid cross, maybe due to mutation, maybe due to polyploidy, maybe due to environmental conditions. So that cross can be modified into another ratios like 9 is to 7, 9 is to 3 is to 4, 12 is to 3 is to 1, 13 is to 3, 15 is to 1, or 9 is to 9 is to 6 is to 1 in different crops. So these are the modified ratios, and this type of ratios is comes under the gene interaction. So maybe 9 is to 7 is called as 9 is to 3 is to 4, 12 is to 3 is to 1. They are maybe complementary, supplementary, inhibitory gene interaction. In this way, these are the various types of modification ratios which comes under gene interaction. Next, that is the pleiotropic gene effect. Pleo means more. So this defines that when one gene was found to govern the expression of two or more characters, means here the single gene is governing two or more characters. One of the best example that white eye allele in Drosophila. But in case of Mendel, Mendel observed and stated that one gene controls the expression of only one character. But there may be chances that one of the gene can express or can govern two or more characters. That is called the pleiotropic gene effects. Okay. So best example that is the white eye allele in Drosophila. Uh, this white eye allele affects eye color, shape of spermatheca, fecundity, and testicular membrane. Means it is affecting more than two characters. It is called as the pleiotropic gene effects. Next, that is the polygenes. So this Nielsen L observed that some characters are controlled by several genes. Okay, and such gene has additive effect in expression of characters. Poly means more. So some of the characters, this is just opposite to the pleiotropic gene effect. So in this polygenes, one character is controlled by several genes. That's called the polygenes. And this concept led to the foundation of polygenic inheritance. Next concept that is about the environmental effect. We all always observe that when there is offspring, epsilon offspring, so we are always observe that the effect which the offspring got, the effect may be from the parents only, okay, or the genotypes only. But in some cases, it may be due to environment also. So some of the effect which the offspring got can be from environment. So gene can interact not only with other genes but also with environment to produce the final phenotype. Means in case of the final phenotype or the final offspring, there must be the sum of the environmental effect, there must be the genotypic effect. So thus phenotype is the result of interaction between genotype and environment, right? So this is about the environmental effect. And the last that is about the maternal effect. So Mendel did not observe any difference between direct and reciprocal process. In the picture, we are observing that some of the crosses, in case of pale green leaf plant, we are considering male. Cross with dark green leaf plant, we consider female. We observe that F1 is dark green leaf plant. Again, if we shuffle it or revised it, so we are considering dark green leaf plant male crossed with pale green. So we observe that the F1 is the pale green. So in this, we can observe there may be two inheritance or two effects. That is one of the effect from the nuclear gene and another is effect from the cytoplasmic gene. So cytoplasmic inheritance, also known as the maternal inheritance, means this cytoplasmic inheritance or this effect we are observing, it may be from the maternal side. That's why this cytoplasmic inheritance, also known as maternal inheritance or extranuclear inheritance, means the inheritance or the effect does not come from the nuclear genes. It comes from the cytoplasmic genes. That's why this cytoplasmic inheritance, also known as maternal or extranuclear, is a phenomena referring to any traits that is not inherited from nuclear genes. Okay. So after the Mendelism, later investigations revealed that the presence of significant difference in the reciprocal process which lead to the concept of cytoplasmic inheritance. These are the paternal effects. So these are the various post-Mendelian concepts which were derived or investigated after the Mendelism. So this is about the post-Mendelian. So thank you. Stay tuned and subscribe to Sage University.